Thank you for joining us today. Here, my colleagues and I will walk you through a series of case studies that highlight how metabolomics can reveal actionable insights for your COVID-19 research. A better understanding of SARS-CoV-2 is needed to develop effective treatments. Currently, there are no antiviral therapeutics targeting human coronaviruses and very few effective treatment options. There is strong evidence that SARS-CoV-2 drives metabolic perturbations that provide growth and survival advantages to the virus that can be exploited to unlock new and effective therapeutic strategies. Metabolomic profiling of SARS-CoV-2 related samples is a vastly underexplored area of investigation with enormous potential to increase our knowledge base of viral biology, mechanisms of infection, and possibly treatments. By measuring metabolites, metabolomics integrates cellular gene expression, transcription, and protein production to assess health outcomes and severity of disease, particularly relevant to understanding the trajectory of COVID-19 infections. Metabolon is the industry leader in the use of global metabolomics and lipidomics to reveal metabolic perturbations across all biochemical pathways to assist with biomarker identification and mechanistic understanding. Understanding the pathophysiology of and the immune response to infectious disease is vital for discovering effective treatments. The following two case studies address several problem-oriented metabolomics applications for SARS-CoV-2 research. In this study, global metabolomics were used to identify lipid metabolites which changed in alveolar type 2 epithelial cells during influenza infection. The method here was isolation of AT2 cells from mice that were infected with H1N1 influenza to induce acute respiratory distress syndrome. So like influenza, severe COVID-19 infections can lead to respiratory failure from acute respiratory distress syndrome. And this respiratory failure from ARDS is associated with increased mortality. Global metabolomics were performed on these isolated AT2 cells. This is important because these are the primary site of infection for influenza virus. And acute respiratory distress syndrome in influenza infection is associated with significant alterations in AT2 functions. So the investigators were wondering if there would be metabolomic changes that were also associated with ARDS in these cells. So here you can see that infection resulted in significantly reduced levels of several surfactant phospholipids, including phosphatidylcholine and phosphatidylethanolamine, as well as increases in cholesterol. Collectively, these can result in alterations in surfactant. And when surfactant is changed, those can lead to increased surface tension in the cellular membrane, changing the functionality of the alveoli. There were also decreased CDP-D choline and CDP-ethanolamine in these cells, and increased phosphoethanolamine and diacylglycerols. These can be associated with changes in phospholipid synthesis. All of these changes result in significant alterations in AT2 function, which can lead to acute respiratory distress syndrome. So in this study, the investigators were able to identify potentially actionable alterations in the lipid metabolome that is associated with acute respiratory distress syndrome during an upper respiratory infection. In this study, global metabolomics were utilized to identify predictors of disease severity and the need for ventilation during respiratory infection and bronchiolitis in infants. The severity of bronchiolitis varies significantly among infants, and it's thought that the interactions among the respiratory viruses that infect the infants, the upper airway microbiome, and the host immune system could impact the severity of the disease. So, utilizing the Mark 35 cohort of just over 1,000 infants who had been hospitalized for bronchiolitis, the investigators obtained nasopharyngeal aspirate samples to have both metabolomics and metagenomics run to identify markers of the severity of infection and the need for positive pressure ventilation. So positive pressure ventilators are increasingly in demand to treat the most severely infected of COVID-19. And so here, another respiratory infection may serve as a model to help understand the interplay of the various factors that would cause the need for this intervention. So using the NP samples, Metabolomic analysis identified biochemical markers associated with a higher risk for severe infection, which are shown in the red, and a lower risk for severe infection, which are identified in blue. So you'll see that some of the biochemicals that were associated with the more severe infection include sphingomyelins and sphingosine, while things like inflammatory lipids were negatively associated with disease severity. Interestingly, a series of microbiome-associated metabolites were also identified in this analysis. These could also be used to predict the need for positive pressure ventilation in the infants. So further, the metabolomics and metagenomic data were integrated to correlate the members of the nasopharyngeal microbiota with the metabolites associated for positive pressure ventilation. So what you can see in A and B panels are that Streptococcus is pneumoniae abundance is positively correlated with the metabolites that were most highly correlated with disease severity. So here, metabolomics of the samples taken from the site of infection were able to identify those who were at risk for the most severe infection, and they can be integrated with other omics to make predictions for the need for positive pressure ventilation as an intervention and disease severity.
In the wake of the escalating global COVID-19 pandemic, the need for rapid development in vaccinology has never been greater. When dealing with a pandemic like COVID-19, vaccine development and discovery of therapeutics is essential to improving outcomes. These next two case studies address the metabolomics applications and Metabolon's capabilities, which are uniquely positioned to address the need for actionable insights for vaccine development. Further, to provide a comprehensive view for more informed decisions to drive operational and quality improvements. This case study talks about how metabolomics can be used to identify ideal media composition for vaccine production optimization. The objective of this study was to elucidate virus-mediated changes in host cells and interrogate the pathways and mechanisms involved in infectious bursal disease virus replication. Infectious bursal disease is one of the most economically important diseases that affects commercially produced chickens worldwide. Although largely controlled by vaccination, new virulent strains of the virus still remain a threat to the poultry population. The challenges, as are with any bioprocessing approach, is upscaling production of the vaccine or biologic or metabolite once developed. Scaling up vaccine production is not necessarily a linear process. In this study design, DF1 cells, which are a continuous chicken embryonic fibroblast cell line, were run across a time course either in an uninfected or infected state. Comparing those infected versus uninfected resulted in profound alterations in a number of different nodes of metabolism. First, in central carbon metabolism, looking at glycolysis in the TCA cycle, it appears that glycolytic intermediates were increased, whereas TCA cycle intermediates were decreased. Additionally, an adjacent pathway off of glycolysis, the pentose phosphate pathway, saw increased carbon input from the glycolytic pathway. The importance of this is that the pentose phosphate pathway, one of its main roles is to generate substrates, precursors for nucleotide biosynthesis. Now, if you imagine when a virus enters a host cell, it basically hijacks the machinery to try and replicate itself and infect the host. And a virus, imagine what it's made up of. In this case, in IBDV, it's a RNA genome, similar to COVID-19, and it's encapsulated by a protein. And when it replicates, it needs to transport from cell to cell. So it needs vesicles, which are made up of lipids. And in this case study, we saw increases in a number of amino acids, which make the proteins for the capsid protein. We saw increases in a number of purine and pyrimidine metabolites, those nucleotides and nucleosides, which would be needed for generating and replicating that RNA genome. We also saw increases in a number of lipid components, which would be needed for that vesicle formation and transportation of the virus within the host cell. Taking what we learned from the metabolomics and marrying that with a design of experiment methodology, the researchers were able to come up with a strategy to formulate a new optimized media. The options for optimization could include things like glucose or lipids, amino acids, nucleosides. And because glucose uptake limitations were apparent and lipids are not the most bioavailable metabolites in cell culture, they focused on amino acids and nucleosides. So applying this to the mathematical methodology of DOE strategy, they developed a model which gave them actionable opportunities for an empirical test of their media. And again, based on the metabolomics data and the DOE strategy, they were able to generate an optimized media, which was able to give them a 20-fold increase in titer. Now, if you imagine a 20-fold increase in titer saves not only money, but time, which is of the utmost importance in the present COVID-19 pandemic. The lessons learned from this example, which is from a chicken virus, hold true for human infectious diseases and will no doubt play a crucial role in the understanding of human viruses and vaccine development, particularly with respect to COVID-19. In addition, Metabolon has experience in vaccinology and the effects of virus infection in other viruses such as herpes, rabies, and bacterial infections such as anthrax and tuberculosis. This case study talks about identifying a limited nutrient in order to optimize a platform process. The challenge in this one was the need for an optimized production process. Comparing it to the needs of today, of vaccine production, the ideology holds true. In this case, it was a large pharma working on the optimization of a platform fed batch process for a therapeutic monoclonal antibody, and they used a GS-CHO system. Similarly, in present-day situations, using a cell line for the virus replication in a COVID background, the ideology of limiting nutrients which could be supplemented to optimize a process, the same ideology is there. The metabolomic insight that was generated from this came from analyzing both cell pellets and spent media. The global metabolomics library that was used to generate the large amount of data could be distilled down to a number of key observations. The first being membranes of cell leakage, if you will. In the media, we identified metabolites such as glucose 6-phosphate, which should not be in the media in a cell culture. When glucose is phosphorylated, it typically is retained within a cell, and when it is seen in the media extracellularly, it is an indication of leakiness and unhealthy cells. Interrogating the different metabolites which were found to be depleted over time, choline stuck out as a key marker. Choline is a very crucial phospholipid head group component, a precursor for phospholipid biosynthesis, and in a bioprocess cell culture, where you're asking a cell to replicate as fast as possible and produce a protein which must be transported, which requires more lipids, choline depletion can have a drastically negative impact. Based on the metabolomics and the recommendation to the researchers to supplement their media with choline, they were able to increase their monoclonal antibody titer. Not only that, the percentage of aggregates was found to be decreased, which is an indication of improved quality. Optimizing this media and product yield allowed for saving time and money. As mentioned before, the lesson learned from this example of a monoclonal antibody can easily be transferred and relatable to virus replication and bioprocesses and vaccinology applications for any virus, particularly relevant to the present day COVID-19 pandemic.
As with other infectious disease challenges, COVID-19 requires researchers to effectively stratify patients for potential therapeutic intervention and perform appropriate risk assessment. Understanding the effects of age, gender, and comorbidities on the susceptibility, treatment response, and outcomes related to COVID-19 are vital. Metabolomics offers a tool to assess patients that may be most at risk. The following 2 case studies highlight this solution. Tuberculosis is among the deadliest infectious diseases on Earth. Nearly 1/4 of the world's population are believed to be latent carriers of M. tuberculosis, and currently there is a lack of tools available to identify those patients who will progress to active disease. Metabolomics is often used in biomarker discovery for non-communicable diseases, but until recently, this approach has not been applied in the area of infectious disease. In this study, global metabolomics was used to identify a group of metabolites that could predict progression of tuberculosis. The GC6074 consortium collected samples from HIV-negative individuals who were household contacts of TB cases and healthy at baseline. Samples were also collected from subjects at 6 and 18 months post-exposure. Integrating Metabolon's global metabolomics platform with machine learning algorithms, a panel of risk-associated and disease-associated metabolites were identified. Disease-associated metabolites, such as cortisol and glutamine, were those metabolites whose absolute difference in abundances between progressors and controls increased on the road to the clinical manifestation of TB. Risk-associated metabolites, such as cotinine and mannose, indicate a higher likelihood of progression to TB. Shown here are the metabolites that changed in progressors leading up to tuberculosis diagnosis. Not shown are the area under the receiver operating curves, demonstrating the significant predictive power of 10 metabolites to distinguish between progressors and non-progressors. These graphs can be found in the corresponding paper. This study demonstrates the ability of using metabolic signatures to detect disease in asymptomatic patients. This suggests that the metabolic changes that occur leading up to the clinical manifestation of TB can be harnessed to identify those patients most at risk of progressing to the clinical TB. Furthermore, this consortium consisted of 4 different collection sites across Africa, providing evidence that this metabolic signature is applicable across lifestyle, genotype, and collection differences. Upon further validation and refinement, a targeted metabolite panel that can be performed routinely could be created. Metabolon's targeted assay team can help customers develop such a panel. Identifying high-risk individuals using this blood metabolite panel could provide enormous value both in recommending therapeutic strategies and in reducing costs of conducting clinical trials that test new treatments and interventions. Additionally, subclinical signatures from a blood sample can aid enormously in controlling an active pandemic. One of the challenges that has arisen with the COVID-19 pandemic is the lack of risk mortality information and patient stratification strategies, particularly in the elderly population, where hospitalization, ICU, and case fatality rates are higher. Low-grade chronic inflammation has been associated with many diseases of aging, but the mechanisms responsible for producing this inflammation remain unclear. Inflammasomes can drive chronic inflammation in the context of an infectious disease or cellular stress, and they trigger the maturation of interleukin-1 beta. This group set out to investigate the role of inflammasomes in the context of human aging. The Stanford Ellison longitudinal cohort was used to combine gene expression with metabolomic data to understand inflammasome activity in vivo. Subjects greater than 85 years of age were assigned into inflammasome module high, or IMH, or inflammasome module low, or IML, groups based on age-related gene expression. These gene expression groups also demonstrated differing metabolic profiles and outcomes. Those with constitutive expression of IL-1 beta nucleotide also exhibited nucleotide metabolism dysfunction, elevated oxidative stress, high rates of hypertension, and arterial stiffness. Those without constitutive expression of IL-1 beta lack these characteristics. Adenine and N4-acetylcytidine, both nucleotide-derived metabolites that were detectable in the blood of the IL-6 beta expressing group, were then found to prime and activate the NLRC4 inflammasome, induce production of IL-1 beta, activate platelets, and neutrophils, as well as elevate blood pressure in mice. These data suggest that the metabolites play an active role in promoting systemic inflammation. Furthermore, in individuals over the 85 years of age, the elevated expression of inflammasome gene modules was associated with all-cause mortality. Thus, targeting inflammasome components may ameliorate chronic inflammation and various other age-associated conditions. A very interesting finding of this study is that the metabolites not traditionally linked to inflammation may be more closely associated to inflammatory responses than originally thought. This raises the possibility of using these signatures as measures of the inflammatory response. More studies investigating the link between metabolites and inflammation are certainly of research interest. As we face the global pandemic that is COVID-19, everyone is looking for ways to contribute. Metabolon is no different, and we have worked to identify areas where we can help. The following case study exemplifies how global metabolomics can provide deep insights into understanding how viruses impact host metabolism. As illustrated here, this understanding can lead to the identification of novel therapeutic targets. Additionally, this case study demonstrates the utility of metabolomics in confirming target engagement and monitoring therapeutic response. Here's a brief overview of what happens when viruses infect the nasal epithelium and respiratory tract. Viral uptake can occur via endocytosis or macropinocytosis. A drop in the pH leads to viral uncoating, and the negative or parental RNA strand is replicated, and the protein-making machinery in the cell is hijacked to translate the RNA into structural and non-structural proteins. The virion is then assembled and packaged prior to cellular export via cell lysis. Rhinoviruses frequently cause the common cold, and effective and practical treatments remain elusive. The current study was undertaken to investigate the impact of rhinovirus infection on cellular metabolism in vitro. Initial studies in cells showed that rhinovirus infection enhanced glucose uptake and expression of glucose transporters. This led the researchers to wonder what the metabolic fate of glucose was once inside the cell. Working with Metabolon, this group of scientists defined the study parameters to answer this question. In this case, there were two stages of investigation. Infection with rhinovirus alone to understand the metabolic impact of viral infection on host metabolism, and then rhinovirus infection combined with 2-deoxyglucose treatment to determine 2-DG's ability to reverse the metabolic impacts of viral infection. Let's take a look at the metabolic data from the initial experiment first. Here is a visual representation of the alterations in sugar metabolism in response to rhinovirus infection. In this fireworks plot, the colored circles represent statistically significant changes. Red circles represent metabolites elevated following viral infection, and blue circles are metabolites that declined. The size of the circle represents the degree of fold change. As you can see here, we have increases in glycogen and nucleotide sugar metabolites, suggesting increases in glycogenolysis. The heat map on the left shows changes in glycolytic metabolites in response to viral infection. Not shown here, but also observed, were shifts in fat utilization and nucleotide levels that may support viral replication. From these data, it was concluded that viral infection led to profound shifts in glucose metabolism, thus generating the hypothesis that blocking glucose metabolism through glycolysis may impact viral replication. To this end, the scientists decided to treat infected cells with 2-deoxyglucose, or 2-DG, which inhibits the conversion of glucose 6-phosphate to fructose 6-phosphate, thus preventing further metabolism. The associated metabolic profile was then examined. Here are the initial results from manipulating glucose availability to infected cells. Both glucose deprivation and 2-DG treatment reduced viral replication. The next slide will show you the metabolic signatures of 2-DG treatment in the presence of rhinovirus infection. 2-DG rescues the viral-induced metabolic reprogramming. Again, we see the data presented in a fireworks plot and a heat map. we see declines in nucleotide sugars along with increases in glycolytic metabolites upstream of 2DG inhibition. These data suggest that 2DG prevents the virus-induced shifts in the metabolism of intracellular glucose. Of course, these observations were seen in an in vitro system, so the scientists then took these results and tested the hypothesis that 2DG may be able to treat rhinovirus infections in an animal model. To this end, rodents were infected with rhinovirus and treated with either PBS or 2DG. Viral replication, immune cell infiltration, and lung pathology was then investigated. These data demonstrate that 2DG reduced rhinovirus replication and immune cell infiltration in the lungs. Additionally, as you can see in the images on the right, the 2DG significantly reduced inflammation in the lungs compared to the placebo-treated mice. This study is a nice illustration of how metabolomics was used to identify alterations in virus-induced cellular metabolomics. Rhinovirus orchestrates the anabolic reprogramming of the host cell metabolism to support viral replication. 2DG was found to reverse these alterations. Metabolomics was important in A, identifying these shifts, and B, revealing a potential novel therapeutic target. Additionally, there was a demonstrated utility of metabolomics to track the efficacy of the potential treatment, in this case, 2-deoxyglucose. Furthermore, targeting glycolysis in this way to treat viral infections, particularly COVID-19, may have some legs. Recently, a biotech company in Texas, Metabolon, announced that a prodrug of 2DG shows some efficacy to reduce coronavirus replication.
Comprehensive metabolomic profiling can simultaneously inform on several biological processes relevant to infection, such as inflammatory and redox markers, energetics, microbiome contributors, and the xenobiotic impact. These insights can lead to predictive response signatures and therapeutic strategies. The following COVID-19 case study, run by Metabolon and our affiliate Dion in China, exemplifies how metabolomics can provide unique biological insights supporting the profiling of infectious disease patients. The study examines the need to better understand SARS-CoV-2 to develop effective treatments. The Metabolon solution delivers integrated understanding of viral activity through metabolomics. For this preprint study, serum samples were collected in Taizhou, China, between January and February of 2020. The study population included 25 healthy controls that were age, sex, and BMI matched to the other individuals. There were 25 patients who had regular SARS-CoV-2 infection, and 21 that had severe symptoms. 25 disease controls were also included in this study. These patients had an unrelated respiratory disease with similar symptoms to SARS-CoV-2. In Figure 1, you can see the sampling scheme for this study. The individuals are broken out as non-severe or severe for the COVID symptoms, with age and sex shown on the axes. Proteomics and metabolomics data were sampled at various time points indicated by the red and blue circles. In panel A, you can see the coefficient of variation for both the proteomics and metabolomics data, which were derived from the QC standards run in each method. Proteomics identified 791 proteins, while global metabolomics identified 847 different metabolites, excluding the xenobiotics. You can clearly see, using the uniform manifold approximation and projection method, that metabolomics is able to identify and separate the COVID-19 patients from those that had the other respiratory infection or healthy controls. In panel A, you can see a heat map of the serum metabolites that were identified in the 4 different groups of subjects in this study. These include metabolites from the fatty acid, steroid, glycerophospholipid, sphingolipid, choline, and tryptophan families. On an individual metabolite basis, you can see that metabolites like mannose and kynurenine follow a similar pattern, where both the non-COVID respiratory infection and the severe COVID respiratory infection result in increases in those compounds in the blood, whereas arachidonate, a polyunsaturated fatty acid, is significantly lower in the severe COVID-19 patients compared to all other groups. So using serum metabolomics, the researchers were able to identify profound alterations in amino acid metabolism, inflammatory mediators, and glycogen synthesis metabolites. These metabolites can be associated with macrophage dysfunction, inflammation, platelet function, and clotting factors, all which may contribute to the pathophysiology associated with severe COVID-19 infection. Key proteins and metabolites were integrated to characterize severe COVID-19 patients in this working model. SARS-CoV-2 may target alveolar macrophages via the ACE2 receptor, which induces pro-inflammatory cytokines such as IL-6 and TNF-alpha. This leads to an increase in acute phase proteins, which are tracked by alterations in metabolites associated with macrophage function, inflammation, and the immune response. Metabolomics and proteomic data were integrated to develop a model to predict severe COVID-19 infection. This shows the study design for the machine learning-based classifier that was used for the COVID-19 patients. Using the training cohort for the proteomic and metabolomic data, the classifier was then validated in an independent cohort. Random forest analysis identified the key proteins and metabolites that differentiated those with severe COVID-19 infection from those with non-severe COVID-19 infection. For example, the protein CRP was a key differentiator, while the metabolite kynurenine was also a key differentiator between those two groups. In panel G, you can see the training cohort clearly separates, and then in the validation cohort, you see the same separation, suggesting that these are useful markers for predicting severity of the COVID-19 infection. In conclusion, serum metabolomics can be used to differentiate COVID-19 infection from other respiratory infection in healthy controls. Changes in metabolites correspond to alterations in inflammation and the immune response. Integration with proteomics allows for the development of a model to predict which infected patients will develop severe disease.